quickly I am going to some transfection experiment. What is that? These are also can you hear me clearly?
Yes, sir.
Okay. So now, so what is the transfection experiment? Now, see, these are the proof that a DNA are the genetic material. The transfection experiment is saying that if we are applying some enzyme, okay, so that the covering outer boundary is removed. So then what will be there? We'll get the protoplast or spheroplast. So this kind of structure we will get if we are using some enzyme known as lysozyme. If we are treating the bacterial cell E. coli with lysozyme, so then we will get that protoplast only. Okay. Now, why I am saying so? Because now if we are infecting those kind of protoplast with some phage DNA, purified phage DNA, okay, so we will find that bacterial cell is transformed with that particular phage DNA. So which is a direct proof that transfection is happening. Transfection is happening. Okay. Just note down that transfection experiment is performed by the application of lysozyme to the E. coli cell and then infecting the that protoplast by phage DNA, viral DNA. We are infecting that particular bacterial cell and found that the bacterial cell is transfected by that particular viral DNA. Is it okay? So directly you can prove that yes, a transfection occurs. Similarly, some indirect evidences are also found which proves that DNA as a genetic material. For example, you can use the mutagenesis effects. So you know there are some rays which are used for making the DNA mutated. So that means if we are applying the UV light or any kind of ray, X-ray or gamma ray, so that the material may be mutated. The genetic material may be mutated. Okay. Now, if you can follow the graph. Is it visible? The graph is visible. The right hand side. Okay. So now, so there may be two options. So you can apply the UV light and you can study the mutation frequency in that particular DNA. Okay, so you can see that the graph can be made like this way, the first graph. So where you are applying the UV light and you are measuring the mutation frequency. So you are getting that at 260 nanometer, the mutation frequency is maximum. Okay. On the other hand, if you follow the absorption property of such UV wavelength, say for example, if you apply this 260 nanometer of UV light, so you will find that for nucleic acid it is maximum. You can see that for nucleic acid the absorption is maximum, and for protein also you will find that you need to apply 280 nanometer UV light, not 260. So the bands are different. So you'll get different absorption spectra for protein and nucleic acid. But if you overlap this graph with this graph, so you will find that only for DNA or nucleic acid, these kind of graphs are overlapped. So that means these action spectra, which is the equivalent to mutation frequency, and the absorption spectra for at 260 nanometer, those are overlapping, but that is not true for protein. Okay, so if you are applying 280 nanometer of UV light, protein is absorbing that particular wavelength of UV light, but that is not inducing any such mutation in inside the amino acids or protein sequence. So that means it is indirect evidence that mutations can happen inside the nucleic acid only, not in the protein. Okay, these are indirect evidence. And another direct evidence is recombinant coming from recombinant DNA studies. So in case of biotechnology, you will find that such kind of recombinant genetic engineering is possible, and such kind of experiment is direct proof that you are extracting some DNA from one organism and you are putting that inside a vector, and then that vector is infecting to some other cells, and that cell is transformed. So that is a direct proof that DNA can be acting as a genetic material, where you cannot use protein to transform one particular cell by transformation principle. Okay. All right. So then you will find that some RNA can be also used as genetic information for some viruses. So for example, TMV, tobacco mosaic virus, is a RNA virus. And in this context, I can say you that what about the nature of coronavirus, COVID-19? So what is the nature of this particular virus? Whether it is a DNA virus or RNA virus. Hopefully we have heard about that.
It's a RNA virus.
Good. So I'm not going to RNA because in your syllabus only the DNA structure is very much important. So again, we are going back to that. Nucleic acids, we have already discussed that phosphate group, 5-carbon sugar. And can anyone recall that why I told you that the sugar carbon is marked with 1', 2', 3', 4', and 5'? Why it is so? No? You cannot, so then tell me. I will explain it again.
No, sir, we cannot recall it.
So the answer is, see, in case of the sugar molecule, the carbon is present, and again, here the carbon is present, the bases, right? So how do you differentiate which carbon it is? If you are seeing this structure on strand, so carbon is present in the sugar molecule as well as this nitrogenous bases. So how you can differentiate it among these two carbons? So that's why in ribose only we are placing one prime symbol, 1', 2', 3', 4', 5', but not in the nitrogenous bases. Is it clear now?
Yes, sir.
Okay. And then I have also mentioned the structure of this difference between this ribose sugar and deoxyribose sugar in place of 2' position. And then the nitrogenous bases, there are purine bases, pyrimidine bases. I told you to remember the structure as well. What are the components of DNA and RNA, particularly in terms of uracil and thymine? What is the difference? and the structure of purines and the pyrimides, a single mm, ring structure and fused ring structure of purine. Okay. And then how the nucleotides are formed. So in case of pyrimidine nucleotides, we will call it as deoxythymidine monophosphate or deoxycytidine monophosphate kind of thing. Whereas in purine nucleotide, we will name it that deoxyadenosine monophosphate, that is DAMP, or deoxyadenosine monophosphate, that is deoxyguanosine monophosphate, that is GMP. So now you can understand that how we can derive the ATP structure from this deoxyadenosine monophosphate. So if we add 2 more phosphate to it, it will become the ATP. Okay. And here is a single strand. We have discussed that how we can derive this 5' phosphate to 3' OH group of a single DNA strand. So that is always forming from 5' to 3', not the reverse. You cannot generate one strand from 3' to 5' end. Always DNA synthesized from 5' to 3'. Okay. And this is a very known phase. Can you know them? Can you name them? Who are them? They are the pillar for solving out the problem of DNA double helix structure. So this
The
is
first one is Watson and Crick. First two.
This is Maurice Wilkins, and this is Watson and Crick, and this is Rosalind. Okay.
Okay.
These are the 4 pioneers who worked on DNA double helix structure, but we often credited these 2 men, Watson and Crick, but sometimes we fail to give the importance of Rosalind Franklin contribution to this DNA structure. So actually, you may have heard about when Francis and Crick was trying to solve this problem, that how this DNA strand is forming this duplex. They are not succeeded to give the detailed result or exact result why this is happening. And at that time, Rosalind Franklin also doing the same kind of job, and she was applying the X-ray crystallography structure. You know what we mean by X-ray crystallography structure? We are applying some X-rays on a particle, and then we are on the X-ray film, we are trying to observe the structure, how it looks like. Now, if we are getting such X or cross mark like structure, so it is very obvious that the structure is forming some helix like structure. Okay, so that is a property form which can be revealed by X-ray crystallography structure, and Rosalind Franklin was doing that. And with the help of Rosalind Franklin study, Francis and Crick proposed the DNA structure, but in the meantime, Rosalind Franklin died, and Watson and Crick got the Nobel Prize for this remarkable experiment and remarkable discovery of DNA double helix. Okay, again, this is the DNA double helix structure which was confirmed by X-ray crystallography structure, and here is a very known DNA duplex which is helix in nature, helical in nature. You can find that. And interestingly, if you try to identify how many DNA strands are there within one turn, okay, within one turn, so you will find that there are 10 such base pairs in one turn, and which is spanning the region 3.4 nanometer. And if you calculate that how many, so you can calculate that what will be the difference between 2 such base pair. If there is a 3.4 nanometer spanned by 10 base pairs, so obviously within one base pair, you are getting 0.34 nanometer, isn't it? Okay, before going to further details, these chemical bonds are very much important in DNA structure. You can study this in detail. Those are covalent bonds, okay, which is forming the base, nitrogenous bases or sugars. So this kind of where the sharing of chemical sharing of electrons are happening inside the bonds. So forming the covalent bonds is very much strong bond, and some bonds are known as phosphodiester bond. I explained it in the last class also how these phosphodiester bonds are formed. This is also example of covalent bond and is a strong bond. Then another type of bonds is known as hydrogen bonding, that is in nature weak bond between an electronegative atom and a hydrogen atom, but in a cumulative way, these hydrogen bonds are making this DNA structure is very strong. That should be remembered, that individual hydrogen bond is weak in nature, but when we are talking about multiple numbers of hydrogen bonds, so that becomes more rigid, more strong in nature. Another kind of bond which is stabilizing the DNA structure is hydrophobic bonds. Okay, so how these hydrophobic bonds are created out? So association between the non-polar groups with each other. So now think about this when. So before going to further details, can you tell me what is the backbone of this DNA? Which part is backbone?
So phosphate, backbone.
Only the phosphate is in
And
backbone?
histone.
Histone? No. Histone is
Yes,
not
yes.
there in
No,
DNA
no,
double
no, no.
helix.
So the ribose sugar nitrogenous group and the phosphate group.
No, the nitrogenous bases are also not in the backbone. I told in the last class also, only the sugar phosphate, that is in the backbone of the DNA, and the nitrogenous bases are kind of thing which is which are what I say, which is attached to this backbone, okay, which are attached to this backbone, nitrogenous bases like this. So you can see the here in this diagram that nitrogenous bases are attached with this sugar phosphate backbone, and those bases are interacting when there is another strand in opposite direction, which is making a hydrogen bond, and this region is hydrophobic in nature. So remember that thing. The interior portion of the DNA is hydrophobic in nature, but the outer side is negatively charged. So that's why outer side can interact with the histone molecule, which are positively charged. Is it clear?
Yes, sir.
Good. So why I am telling so? Because DNA structure is totally dependent on these characteristics, and so these are very much important. So I have already discussed about the detailed structure of this. So you can see that A, adenine and thymine are forming 2 hydrogen bonds, whereas guanine and cytosine forming 3 hydrogen bonds. So you can understand that inside one DNA, which region will be easily separated out and which region will not be. We need to give more force to separate out the DNA strands. So what do you think? Which region, if it is made up of AT rich region or if it is made up of GC rich region, so which will be easily separated out? If we are trying to
So AC will be easily separated rather than GC.
Right. Because we need to give more force, more energy to separate out to dissolve 3 hydrogen bonds, right, which are present in cytosine and guanine. And that's why you will find that during the starting of replication or any kind of transcription processes, the region where the starting area, mostly those are AT rich. Okay, AT rich. That means we'll find maximum number of A and T at that particular site, so that within very using very little energy, that part can be separated out. Okay. Again, the same thing is represented over here. Now see that. What do we mean? This is a space filling diagram. So in bioinformatics, sometimes you'll find that this kind of diagrams are also there. So this is known as space filling diagram, where you will find that different color spherical or circular symbols are used according to their sizes. So you can see that hydrogen, then oxygen, carbon, carbon and nitrogen in base pair, and phosphorus. So all these are marked by different colors, and thus this is known as space filling diagram. Okay. So sometimes you will find that some ribbon like diagrams are found in case of protein or DNA. So there are different structural views. Okay, and now you will find that the base pairs which are aligned this way, so they are again stacked. So that is known as base stacking. So I'm giving this view. So this is one base, this is another base pair, another base pair. So if you just put one coin after one coin, and then another coin. So if you do this thing, so you will get a stack of coins, isn't it? So just like that, the base pairs are also stacked inside the interior part of this DNA double helix. And due to the helical nature, there will be two groups, minor groups and major groups. I discussed about this, and based on that, there are different kinds of DNA strands possible. So mostly we find B type of DNA, bDNA in our cell, but maybe variation you will find in some cases like A DNA or Z DNA. A DNA is right-handed and B DNA is also right-handed, but Z DNA is left-handed, which is mostly, we know that this is a zigzag orientation. Okay, and if we discuss about the base pair pattern, we'll find that in case of bDNA, it is approximately 10.5 base pair pattern, whereas some more bases is there in A turn, and ZDNA has two more than bDNA. So that's why the ZDNA is lesser in diameter than bDNA, whereas ADNA is much thicker, or diameter is higher than bDNA in case of ADNA. So you can see in this diagram also, this is a B type of DNA, and this is A type of DNA. So if you think minutely, then you can differentiate it from these two type of DNA. Okay. So based on that, okay, so a few things are left. Okay, so now we'll discuss about the properties of such DNA structure, because DNA structure, based on this DNA structure, we can determine or we can analyze the properties of DNA. How we can do that? So one such property is known as TM value. TM value. So usually it is represented as capital T and small m, which is known as melting temperature. Okay, melting temperature. Now what is that? Just now we discussed that if we are trying to separate out two DNA strands within a helix. So what we can do, we can give some energy outside, right? So that may be temperature. If we increase the temperature, so then we will find that some region will be separated out across the DNA. Is it clear? So that means the DNA is melted out, in other terms. Yes or no? Hello? Are you there?
Hello.
Yes. Can
Yes.
you hear me? Okay. So see, now if you are trying to separate out two DNA strands, so what you can observe, this is the property. Now you can see that you are increasing the temperature and you are measuring the OD value. OD value is just the what I say. There is a experiment known as colorimetric experiment. Have you heard about this thing? Colorimeter.
No, sir.
Okay, fine. So you will find in your experimental lab a kind of box like instrument where you will place one DNA and you can measure. You will apply one particular wavelength, and then you will find that how much of that particular wavelength is absorbed by that DNA sample, and then it will give you one particular OD value, optical density value. So optical density value here it is at 260 nanometer wavelength. Okay, it is measuring, it is coming out from that instrument. And if you repeat this experiment at different time point or different temperature, so what you will find that the graph will look like this. Okay, and the midpoint of that particular graph is known as melting temperature. Okay, so that is one experiment for red one, and another experimental result for the green one. Now here it is marked that TM value for this DNA is 83. Okay, and TM value for this DNA is 77. Okay, now I am saying again that melting temperature is one which is defined that when the half of the DNA is melted out. Okay, half of the DNA is melted out. So what do you think? Which one will be melted out quickly? Which DNA? The red one or the blue or green one? Are you getting my question, or yes or no? Hello? G, Toritro, Pratik. Anyone? Are you tired? Is there any connection issue?
Yes, sir. Can you hear me?
Yes, now I can hear you.
Okay, sir.
So can you tell me what is going on here?
Sir, as the temperature rises, the DNA.
सर आवाज आ रहा है अभी आ रहा है
Yes, now I can hear you. So can you tell me what is going on here in these two experiments?
Sir, the first experiment, the molecules are differentiated because of the rising of the temperature.
Mhm.
And in the second one, sir, can you please explain the second one again?
Yes, similarly, we are taking the we are doing the same experiment by using the two different sets of DNA. So this DNA, which shows that the melting temperature for this is 77 degrees Celsius. So that means the half of the DNA has already melted at 77 degrees Celsius. And for this green color, we are getting that for this DNA, the 83 degrees Celsius, at 83 degrees Celsius, half of the DNA is melted out. So it clearly shows that we need to put extra energy in the second case, isn't it? So now you can
Yes.
conclude that which DNA has more GC content from this kind of analysis. Is it very straightforward? From this kind of graph, if you are getting, now from this kind of graph, you can directly conclude that which DNA has more GC content.
Yes, sir.
Can you say that whether this is having more GC or this is having more GC?
So the temperature with 83 degrees Celsius have the more GC content.
Very good. So that is one application of this kind of structural analysis, and the opposite is known as renaturation of DNA. If we again remove the heat, so temperature, so then again the DNA strand will rewind or will be forming the helical again, renature. And the second one is FISH experiment, that is known as fluorescent in situ hybridization. Okay. So that is a very beautiful experiment with some probes. If we are explaining this slide, so here you can get that the probe is specific to the centromeric DNA, and in metaphase stage under the microscope we will see this kind of chromosomal structure. Okay. So red fluorescent is produced by propydium iodide counterstaining of the chromosomal DNA, and yellow fluorescent signal indicating that the centromeric region. So this kind of experiment is performed with the DNA structure. Another is electrophoretic separation of DNA, which is also based on the DNA structure. Because as you are a bioinformatics student, many times you will get that biotechnological experiment that how the DNA strands are separated out, or how the different sizes DNA are isolated from a mixture of DNA. So you will find that there is an experiment known as electrophoresis. Probably you have already seen in class 2 level also. I'm not repeating the same experiment again, but at the experimental part, you will find that some ethidium bromine strained DNA band are obtained, and theoretically the longer fragments will not move faster in this kind of gel, but the shorter fragment will move forward, and you will find such kind of bands in the electrophoresis gel. Okay. These kind of techniques are directly associated with DNA structure. And then comes to bioinformatics part. Now, if you are talking about DNA structure in bioinformatics, so that is, you can view this three-dimensional structure, and if you are considering the sequence, so then your job is to find out the sequences of that particular DNA from a website known as NCBI, or any kind of website you can use. But to know about that particular DNA more, you need to do one basic experiment that is known as BLAST. So I will give you this material, and if you can do, you follow these steps, there are certain exercises how you can do BLAST. So go to that website and click on the nucleotide BLAST, put that sequence. Okay. So you do that experiment, you run the experiment, and just getting to know how you can run BLAST. So this is your home task. And another home task is you need to find out what is the helicity of these two strands. So I am giving you one clue that when you are trying to find out the helicity, whether it is a right hand helix or a left hand helix. So right hand helix means when you just keep your hand in this position, and if you find that the helix is clockwise, okay. Can you get my point? If you are moving this in this way, so helix is clockwise. So that is the right hand helix. And if it is anti-clockwise, if you are using your left hand, so you will get that the helix is left hand orientation. And now your job is to find out from these two diagrams which one is right-handed helix and which one is in left-handed helix, because DNA can be in any form, left-handed or right-handed helix form. So you try to find out that. And for that, you need to consider this line. This is very interesting. Look carefully that this line, this axis is not touch this is. So that means this strand is formed in this way. This helix is going from this direction to this direction. And here the helix, this helix is behind this line, and it is coming upward and then going back. So using this clue, you try to identify which helix is that, whether it is a right-handed or left-handed helix. With that, I am closing this session. Okay, and